Okay, so we're gonna do the last hands-on session for today, which is basically a combination of the last two talks we've heard, using the GUI and using the command line interface. And we're gonna do this again via Jupyter Notebook. I think you all received an email with an invite to the HBP Collaboratory. And I think most of you accepted and created an account with that. Is that correct? Yes. Okay, because that Jupyter Notebook you will find on the HBP Colab. So the easiest thing to do if you want to do this together is just to download the notebook, the Jupyter Notebook.
You didn't invite your colleagues?
I didn't send the email.
Okay.
If not, I can also just send you the notebook if you want to follow along. So you do this. We saw this early in the morning with Petra. She showed us that we are all on there now. So if we go into the HBP Collaboratory, did you all get in there? Did that work? You just go to Collabs and search for TVB, which is the easiest way to find us. If my internet works. There we go. So you just search for TVB. And then we go to TVB Simulator Collaboratory. And we see down here on the left, we see the TVB nodes. And then we see TVB Node 10, which is today. And so far, it's the TVMB notebook so far, but the other ones will come in there as well for the next sessions that we have in the next days. They will all be uploaded over here. So just to get us started, this might look a little bit weird on your screen. I had some issues depending on what browser I was using for it to not show code or some weird displaying issue. But if we just download the file, for you it should have a download sign somewhere here. If you just download the file to your desktop or to your downloads or wherever you want to go, we can use that, which is the easiest way to just do it. Did you all get the notebook? It's in the...
How did you get the TVB from the HBB homepage?
I went over here to Collabs, which gives you an overview of all the collaboratories on the Human Brain Project. And then just filter for TVB. Yeah, it's the tab over here. The other ones found the collaboratory. Right, and then we just go to the TVB simulator again. And we see the hands-on for the TVMB, the virtual mouse brain. Whenever that's loading up. See, some people are still searching for it. You got it? Okay. So, depending on where you started this, or where you downloaded this, we were gonna start the Jupyter Notebook. For this, we just go into the directory where we downloaded our IPython notebook to, which for me is my GitHub. And then I call the Jupyter Notebook to start. So for this, I start on the Jupyter Notebook. And this again should give you an overview of all the Jupyter notebooks that you have in that directory where you called the Jupyter Notebook app. Were you all able to open it? Guess I'll wait a bit. Did you find it, Kurt?
It's under, yeah.
Yeah,
It's trying to open.
If you become a member of the HPP collaborator, you'll also be able to just run it on the collab. But since this is not the case yet, we will have to download and just run Jupyter Notebook locally. All right. Can I go? Did you get it, Kirit? You got it open?
I just found a way to run it.
No.
Keep on.
Okay. So basically what we're doing now is we're trying to, we're actually going to, we're just going to succeed, run a simulation like we did before, like we saw in my first simulation session, but this time based on a connectome for a mouse. And we're not going to simulate EEG data or the subsample. I don't know what you used in the previous one. We're going to try to simulate resting state functional MRI based on our mouse connectome. So there are a couple of steps that we're going to take to run this and to perform some basic analysis on this data. Again, all the analysis is implemented in TVB, so this is what we're going to do here now. We're going to look at the functional connectivity dynamics, and we're going to use the nice visualizations that we have in the TVB GUI. And to make this possible, we're going to have the interaction between the command line interface and getting data from there into the web GUI. We're going to do this while the simulation is running because this is going to take a little bit of time. So the steps that we have is first, we have to prepare a workspace like we did before, where we loaded all the TVB libraries that we needed for the small simulation. In this case now for us, it's going to be a little bit more. So the first step is just we're going to run all these imports, which basically import all the necessary functionality from TVB that we will need, as well as some additional Python packages like NumPy, etc., H5Py for the data formatting that we're using, etc. The next one is just, we will not have to go through those functions. These are just helper functions that we wrote for the data handling later on. We just run it and put them in the workspace. But we will get to the little details of that a little bit later if we save our data. So the first thing that we need to adjust is the data path to where our data is. If you're on Mac, this path should pretty much stay the same depending on where you put your TVB distribution folder. When I downloaded it, I moved it to my applications folder, so I found it under applications, TVB distribution, and then I access the TVB app content. So if you're on Linux or on Windows, that might be a little bit in a different place. So I'm just showing this really quick and how it is in Mac. So we have the TVB distribution folder, and then we access the data that we have in the database, like we saw earlier, where if you have the web GUI, we automatically have our database created with all the default data. We have it in the TVB app, and then we go and look at the package contents. So if I...
One thing for the person who uses Windows, normally you have in the folder TVB distribution, TVB data.
Exactly. This is what we have it. This is exactly this data. This in the Mac version, it's a little bit hidden. This is exactly that data, TVB underscore data. So it's easier to see if we just go this. It's in the TVB distribution folder, in the app contents, and the resources in Python. In the Mac, that's a little bit hidden, and in Windows, it's a bit more obvious to find the TVB data folder. So the easiest way is to adjust this path to the TVB data folder right here, so we can use this later on. You could also just move the data to your desktop or whatever, but it's easiest to just adjust this path so that it's pointing to the TVB data, because in this data folder, what we see with our default distribution of TVB, we see the mouse folder, and we see the LM 2 millimeters, and this is already containing the 4 files that we're gonna need to create our mouse simulation model. You see they're all in H5 file format. We get to this a little bit later, but this is it's a TVB specific H5 file containing all the information that we're gonna need. So what we're gonna do is we're just gonna run the cell again. We've seen this before with the Jupyter node that we just had with Paul. And then the next step is just like with the simulation that we had before. This is should be familiar now. The first thing that we're gonna need with all these simulations is a connectivity matrix, or the 2 connectivity matrices with the weights and the tracks. And this is just happening by loading in our connectivity again from a given file. And for this, we just add our connectivity H5 file to the path that we had. So we are just loading in from the TVB data path the connectivity H5 file. If we also just want to take a quick look at it, we already see this because I've run this notebook before. The structural connectivity matrix for our mouse connector. How that is created, we can look at that in a little bit while the simulation is running and we have basically some free time. We're seeing this is a little bit different than the nicely prepared one we had for the human before, where we got rid of all the interhemispheric connection that didn't matter to us. So now we see a wider range of values, wider range of connections here than we had before with the human simulation that we just ran. So the next step is again to define the model that we want to use. In this case, because we are reproducing the results from a 2016 paper on Velozzi, we're using their models, and which is in our case the reduced Wong-Wang model. I think we've heard that name earlier today a few times, with just the default parameter that we have from the paper to have some sort of biophysiological feasibility of those parameters. And just like in the simulation that we did before, we're defining a monitor being the variable that we want to monitor during our simulation, the output that we want to get. And in our case, this is the bolt signal. And we're recording this every 2 seconds. I think before we had the temporal average every second or every millisecond. And now we're just going for the bolt signal. So we're very slow recording time of 2 seconds, which is the standard that we have in functional MRI. And just like before, we're also defining coupling. Again, we just use a simple linear coupling between our regions in the network and setting the parameter here to 0.5. Those are chosen so when we run this simulation here now, we get some nice looking results. This is not biologically feasible or founded. This is just so when we look at the simulation afterwards, we get a nice looking results now. We see in the brackets behind the feasible parameters that they explored in the actual paper. And again, we're defining a duration of how long do we want to simulate fMRI or simulate fMRI recording, and we're going for 5 minutes here. The standard would be for the resting state MRI would be 20 minutes, but that would take on my computer, which is pretty nice, takes about 47 minutes on average. And I think that is not a time that we just want to sit around and wait for this to finish. So we're going to shorten it to 5 minutes, which take roughly 10 to 12 minutes to simulate anyway. So this will give us some time to do some other stuff in between. And then we're going to combine all of this in our simulator object, like we did before. We say, we build our simulator, we give them the model that we refined, the reduced Wang. We use the connectivity that we gave them, we use the coupling. We use an Euler stochastic integrator this time with an integration step of 0.1 to not make it too small. So it's a little bit faster. And we're adding some noise at each step to make this a little bit more feasible. The monitors are the monitor that we defined, which is just a bolt signal for us. And we give them the duration that we want to do. So we just run this. And also, this is a very important step. We just put this here at the end of the simulator. That was in the one that we did before. This was a single cell where we said sim.configure. I noticed that for a few of you, this led to some problems because we forgot to run it. So this is the very important part to make sure that this simulator actually, the simulator object gets configured. And the next step would then just be to run the simulator object that we just created. And as an output, give us the bolt timing for when we record it and the actual data. So if you just click on run here, we see that we have the asterisk in our notebook and this is running in the background. This will take, like I said, because we're simulating 5 minutes, around 10 to 12 minutes to run, maybe a little bit longer in your computers. And so we basically have to wait. In the meantime, what we're going to do is, because this is trying to a little bit bridge the gap between the computer, the command line interface and the GUI, we're going to go and build our new project inside the GUI for our mouse simulator. So we again going to go back to our TVB GUI that we had before. Do you still remember on how you open that? So try to open it and create a new project. Just again go admin and pass. And we are in our by now well-known and beloved web interface. Did everybody get there? I know one Windows user where this will take a while. Everybody else. Okay, so I'll just go ahead and we're gonna go again on the project tab to create our own little mouse project. So to do this, we go again to our overview of list of all project and we click on this nice little star here on the right that says create new project. And we call it my mouse simulation and save. So, cool. There we go. And now we see our new project, my mouse simulation. It's selected by default. And we see 0 bytes because there's nothing in there yet. So what we want to do now is, while the simulation based on the connectivity is running in the background, we want to load the data that we have for our mouse into our project so that at the end we can use the nice visualization, the nice 3D visualization of our simulation at the end. So if we go into our project and we go into the project data, we see that there's nothing there. Again, we go to the right, we see the operation available, we say upload our data, and then we come to this overview of all the different data types that the TVB is able to process. And it's like Paul said before, even if we are loading in connectivity data, this can come as a CSV file, a text file. We can upload structural MRI images in nifty format, in GIFTY format surfaces if we're using nice free surfer. All kinds of different appropriate file formats, but for us, because we have this default mouse data that we just found in our TVB data file, we're going to use the HDF5 file format, the TVB specific, and we're just going to upload the different file formats that we have. An important thing is here, while I'm doing this, I have to look this up myself. For you guys, if you have not created this this project yet, to go into your files. The path is not automatically selected. That is nice. If we want to upload this, that is an error I did not had yet. TVB cannot open the TVB app content path that we defined. If we go back here, where is it? It's here. Our path to the data that we had here. The Jupyter Notebook can open it, the file that we had here, but TVB cannot access the TVB content, the TVB app content. So to upload this data, we will have to copy this file, for instance, to our desktop. So I still have it open here, the LN 2mm folder that we had in our mouse folder in the TVB data. We just copy that to our desktop, not move it, copy it. Copy that to our desktop. Anybody know how to do that on Apple without creating a link?
The whole folder?
Yeah, the whole folder. Do you know?
Just control anything RC and anything
Hmm? No, you can't go like this. It doesn't do it.
What?
Yeah.
Yeah, it does.
But it should do it. I mean, I can't. If I do this, I just do. I just create a link.
What?
I cannot move it. I can move it to him. I just go like this. I copy it. Okay, that works. Weirdly enough. Command C doesn't work. So we have this whole data accessible for the TVB web GUI. Did you all manage to do this faster than me? Probably. Perfect. Okay. So if we go back to our web GUI, we can now actually upload this data. So the first thing we want to do is we want to upload the connectivity file that we have in our LN folder. So we just go, we select LN, we select connectivity, and we want to upload this. For me, this is going to give an error. So here you see I select the file connectivity and I try to upload, and it gives me this nice red color-coded error because I already have this data loaded into my TVB. This is the exact point I made earlier, that this global ID is very important. As you can see, it says this global ID is already present in TVB, so I cannot upload it again. So what I will have to do is what I showed you earlier. I will have to link to the project. The data I have already in here. So you can see the connectivity is already here, and if I click on it, I can tell it, link this to my new mouse simulation project. And it's linked. It's pretty fast. So you would have to upload all the files in a specific order. That is, the first file that we just uploaded is the connectivity. The next file that we have to upload is the volume. If we don't do this in the correct order, TVB will give us an error because, as I said earlier, this is H5 file. So in each of the files, we have the global identifier to the corresponding files. For instance, if we go to the region volume mapping, if we try to upload a region volume mapping, then TVB recognizes that the volume and the surface or whatever in that case is this region volume mapping is connected to is missing. So it won't let us upload the corresponding file. So we have to go step by step, and basically in the way this was created. First we have the connectivity, then we load the volume, then we load the corresponding region volume mapping, and at the end we load the structural MRI of our animal mouse, which is just basically these 5 files. So we go connectivity and upload, volume, region volume mapping, structural MRI. So we don't go alphabetically, but by the way they were created and the integral dependencies of the H5 files that we have in TVB. So I'm gonna just link them to our new project real quick, so that I can access all those files as well. So, but if I go into my list of projects, this is something how this should look like for you. You go in and the data structure is just those 4 files that we just uploaded. Did that work for everybody? Perfect. So, actually, let's close that one. So we can see the simulation is still running. For me, that is the case. Probably take a couple more minutes. Still see some unsure faces. Did most of you get to this point in the GUI that you got all the stuff uploaded? Yes. You can give the number of CPUs used, but I'm not sure how you do that in the local thing right here. It's probably, I mean, you can do it just in Python anyway. If you go via Python threading, you can probably do that, include that, but I don't know how that is included in this one. But I know that if you run it in the cluster, that is possible.
I think Dennis talks about adding when
Yeah.
you do the multi-scale.
Yeah. Otherwise, that will take years to compute. So he'll do that probably more in depth tomorrow. I'll show you where that is done. Is any simulation for anybody else already finished? I guess not. I mean, like I said, I already reduced the simulation time to 5 minutes to make it a little bit faster for the talk today. But we can't make it too short because we still want to look at the analysis afterwards. And if we just simulate like we did before, a couple of seconds of data, we will not find any nice functional connectivity dynamics or functional connectivity. So my simulation is done, but I guess I'll wait a little bit longer. I mean, this was pretty much 13 minutes now just for the simulation. I mean, an interesting thing that we can look at while we're still waiting for more simulations to be finished is just where that connectivity data that we are using in this case is coming from. And I'm pretty sure most of you heard of the Allen Institute, and this is what we're using. This is the data that we're using to create this connectome. And in the TVB, there's functionality included which is called the Allen Connectivity Builder. So what it does is it automatically downloads the high resolution image of the Allen Institute, of the tracing data from the Allen Institute, and computes a connectome based on parameters that the user will give them. This is basically, to make this a little bit shorter, basically how big the regions at least have to be for us to be included. For us, in this case, this was 2 cubic millimeters. So basically a voxel, since we are talking about standard MRI imaging and not high Tesla. And the ratio that we want to see of target regions of the projection and the injection density of the region, and the minimum number of tracks ending in this region for it to be considered feasible. This was, for instance, in our case, this was 50. And there's a pretty nice visualization if you just go to the Allen website and want to actually take a closer look on that data. That link should work. We can go and look at the different regions in the brain that we have and just select, I mean, this is just very random now, in the olfactory nucleus, and then we can see the actual tracing data that this is based on, with the olfactory nucleus as the seed point, and we see the tracing along, you can select this, along the traces that we have in high resolution. And what the Allen Connectivity Builder does is it downloads this data and computes a connectome based on the parameters, like I said, the size of the voxels, the ratios that we want to include in. And this is just the right hemisphere. They just do that on one hemisphere because they assume the high symmetry of the mouse brain. So on the other side, this is just done on the mirror image of those traces that we find here, which I think is a pretty neat feature, actually, if you want to look at some nice data. Did some more simulations finish by any chance? I mean, I can still hear some ventilators, some fans running, so I guess there's still some simulation going on. Still, the next step are basically, since this is a notebook, easy to follow along if you're a little bit behind with the simulation. What we did is we ran the simulation and now created the time bolt and the data bolt variables that contain our simulation data. And what we want to do is, because later on we want to use this in the nice visualizations in the web GUI, we save this time series. And this is just a function that we have that uses the stuff that we put in the helper functions above. And what we give as an input is just the data array, the time array that we have, the monitors that we looked at, which is the bold activity that we saved in our model, the actual mathematical model, which is the reduced 1-1 that we used, and we give them 2 IDs. And those are exactly the global unique identifier that TVB is using for these data files. Like I said, if we're using this H5 file, if we implement and import data into TVB, it needs to know what the corresponding data files are. And in our case, it needs to know, if we want to visualize it, what is the connectivity that this time series simulation is based on, and what is the region volume mapping that this time series is based on. So basically, what are the actual regions that you used, and what is the connectivity strength that you used for this model. And these are the unique IDs that we can just, in this case, they're just hard-coded because they don't change. So we save that time series so we can later on import it again. But for now, we just take a look at the simulated bolt series that we have. It should, again, look something like this, but a little bit slower, shorter, because we only simulated 5 minutes, so 300 milliseconds. And we see basically what you see if you look at either empirical or simulated bolt series. Kind of a mess if you're looking at all the regions. So what we want to do now is we want to look at the actual functional connectivity and the functional connectivity dynamics of this bolt data that we just simulated, and see if we can reproduce functional hubs in the mouse connectome that we saw in the paper. So what we do for this first is we again create the data type of the time series, which is the TVB specific data type. And what we do is we define windows. I think Petra told about this a little bit in her talk this morning. What we do when we look at the functional connectivity, we first compute the correlation between the regions for a given time window, and then we slide this window across time, because it's a time window, with a given overlap that they're having. And then at the end, we correlate the functional connectivity matrices that we have. In particular, it's the upper triangle of the functional connectivity matrix, to see a change or the stability of that functional connectivity over time. So so this is what we're computing here. We give him the window lengths that we say, okay, a 10-second window with a 2-second overlap, and we slide in this window across our time series and compute the functional connectivity matrix for each of those time series, and then use those windows to create the correlation over time, to call the functional connectivity dynamic in that case, which should be pretty quick if we run this. And the next step would be to visualize this. So it's again just plotting those functional connectivity dynamics. So this is basically similar. It's a little bit small, similar to what we saw in the talk from Petra this morning. Now that's a bit too big. Yes, where we can see that over time, we're here at the axis of time, the correlation between the different functional connectivity matrices stays somewhat stable if the time frames are close to each other. So if we're close to each other in time, the functional connectivity is going to be similar. But we also see areas, which is not too clear here, but there's a little bit brighter spots that we can see that even if we differ the time frames, we see a correlation in the functional connectivity. So we can say we have a little bit of stability here, that we can say in these epochs, we see a stable functional connectivity. And this is what's plotted here on the other side, even though my monitor is not showing this very nicely. We see what is a little bit here separated into similar epochs, where we say in this time frame, the functional connectivity stays is similar, so it's stable. We have a high correlation over time of the function connectivity matrices. And the next step would be, I mean, this is nice to have. First of all, it's nice to have the stability over time, but to try to identify hubs in the functional connectivity that we can see over time. So this is what we do with using functional hubs, computing those. This is pretty much standard graph theory approaches using the eigenvectors and the eigenvalues of the functional connectivity matrices. But I think this would be, I have to zoom out again, too much to go into that one now, but it's basically just trying to figure out which regions of the brain are stable in those epochs in the functional connectivity that we see have the high correlation in their activity. So for this, again, we load in the region volume mapping, because like I said earlier, this is where we have the information of what the regions actually are that we're using, and the structural MRI that we have of our brain, of our mouse brain, just as a background for the image, so we can see the overlap of the regions. And if we plot this, we see exactly what we saw below here. A bit different. We see the regions that are stable in the first epoch that we see. So in this time frame right here, the regions that stay highly correlated over time. So are functionally connected would be the interpretation in that for this resting state simulation that we just performed. Again, the biological interpretation of this in this particular case is a little bit in the background because I adjusted the parameters so we can have a nice simulation, even though we're just simulating 5 minutes instead of going for the actual 22 minutes, so we can go to a more feasible parameter selection. And the last thing that we want to do is, like I said, use the nice web GUI where we created our project. So we're gonna go back to our web interface, which is open already here. And we're again gonna upload an H5 file. In this case, it's gonna be the H5 file that we just saved, which is the simulated time series. So we go again to our TVB 85, choose our time series that we just created, which is saved or stored in the same area where you save the Python notebook that we just ran, which for me was somewhere here. Exactly, that's the one I just created. Nope, that's the one I just created. And we're just gonna upload this into our TVB. Again, click on upload. And now we see again our red data file, the time series region that we had when we used the simulation or ran a simulation within the GUI. So basically we're ending at the same point that we did with the simulation before, except that first we ran the simulation in the computer in the command line interface, and now we imported our simulated data, which is the resting state simulation here, into the web GUI, and which means we can go ahead and use the visualizers. And because we told them in the H5 file what is the volume that is corresponding to our simulation, what is the connectivity, we can use the time series volume visualizer and use this to get a nice visual representation like we had that earlier with the human brain, of the mouse brain, of what is the resting state activity is that we just simulated. That's basically it. Did everybody get to that point, or did I lose all of you at some point?